Self. About 14 billion years ago, the universe arose as a cataclysmic explosion of hot, energy-rich subatomic particles. Within seconds, the simplest elements, hydrogen and helium, were formed. As the universe expanded and cooled, material condensed under the influence of gravity to form stars. Some stars became enormous and then exploded as supernovae, releasing the energy needed to fuse simpler atomic nuclei into the more complex elements. Atoms and molecules formed swirling masses of dust particles, and their accumulation led eventually to the formation of rocks, planetoids, and planets. Thus were produced, over billions of years, Earth itself, and the chemical elements found on Earth today. About 4 billion years ago, life arose, simple microorganisms with the ability to extract energy from chemical compounds and, later, from sunlight, which they used to make a vast array of more complex biomolecules from the simple elements and compounds on the Earth's surface. We and all other living organisms are made of stardust. Biochemistry asks how the remarkable properties of living organisms arise from the thousands of different biomolecules. When these molecules are isolated and examined individually, they conform to all the physical and chemical laws that describe the behavior of inanimate matter, as do all the processes occurring in living organisms. The study of biochemistry shows how the collections of inanimate molecules that constitute living organisms interact to maintain and perpetuate life, governed solely by the physical and chemical laws that govern the non-living universe. Yet organisms possess extraordinary attributes, properties that distinguish them from other collections of matter. What are these distinguishing features of living organisms? A high degree of chemical complexity and microscopic organization. Thousands of different molecules make up a cell's intricate internal structures, Fig. 1-1a. These include very long polymers, each with its characteristic sequence of subunits, its unique three-dimensional structure, and its highly specific selection of binding partners in the cell. Systems for extracting, transforming, and using energy from the environment, Fig. 1-1b, enabling organisms to build and maintain their intricate structures and to do mechanical, chemical, osmotic, and electrical work. This counteracts the tendency of all matter to decay toward a more disordered state, to come to equilibrium with its surroundings. Defined functions for each of an organism's components, and regulated interactions among them. This is true not only of macroscopic structures, such as leaves and stems or hearts and lungs, but also of microscopic intracellular structures and individual chemical compounds. The interplay among the chemical components of a living organism is dynamic. Changes in one component cause coordinating or compensating changes in another, with the whole ensemble displaying a character beyond that of its individual parts. The collection of molecules carries out a program, the end result of which is reproduction of the program and self-perpetuation of that collection of molecules, in short, life. Sources: ASPL/Science. Mechanisms for sensing and responding to alterations in their surroundings. Organisms constantly adjust to these changes by adapting their internal chemistry, or their location in the environment. A capacity for precise self-replication and self-assembly. Fig. 1-1c. A single bacterial cell placed in a sterile nutrient medium can give rise to a billion identical daughter cells in 24 hours. Each cell contains thousands of different molecules, some extremely complex, yet each bacterium is a faithful copy of the original, its construction directed entirely by information contained in the genetic material of the original cell. On a larger scale, the progeny of a vertebrate animal share a striking resemblance to their parents, also the result of their inheritance of parental genes. A capacity to change over time by gradual evolution. Organisms change their inherited life strategies in very small steps to survive in new circumstances. The result of eons of evolution is an enormous diversity of life forms, superficially very different, Fig. 1-2, but fundamentally related through their shared ancestry. This fundamental unity of living organisms is reflected at the molecular level in the similarity of gene sequences and protein structures. Source: The Garden. Despite these common properties and the fundamental unity of life they reveal, it is difficult to make generalizations about living organisms. Earth has an enormous diversity of organisms. The range of habitats from hot springs to Arctic tundra, from animal intestines to college dormitories, is matched by a correspondingly wide range of specific biochemical adaptations, achieved within a common chemical framework. For the sake of clarity, in this book we sometimes risk certain generalizations, which, though not perfect, remain useful. We also frequently point out the exceptions to these generalizations, which can prove illuminating. Biochemistry describes in molecular terms the structures, mechanisms, and chemical processes shared by all organisms and provides organizing principles that underlie life in all its diverse forms. Although biochemistry provides important insights and practical applications in medicine, agriculture, nutrition, and industry, its ultimate concern is with the wonder of life itself. In this introductory chapter we give an overview of the cellular, chemical, physical, and genetic backgrounds to biochemistry, and the overarching principle of evolution, how life emerged and evolved into the diversity of organisms we see today. As you read through the book, you may find it helpful to refer back to this chapter at intervals to refresh your memory of this background material. 1.1 Cellular Foundations. The unity and diversity of organisms become apparent even at the cellular level. The smallest organisms consist of single cells and are microscopic. Larger, multicellular organisms contain many different types of cells, which vary in size, shape, and specialized function. Despite these obvious differences, all cells of the simplest and most complex organisms share certain fundamental properties, which can be seen at the biochemical level. Cells are the structural and functional units of all living organisms. Cells of all kinds share certain structural features, Fig. 1-3. The plasma membrane defines the periphery of the cell, separating its contents from the surroundings. It is composed of lipid and protein molecules that form a thin, tough, pliable, hydrophobic barrier around the cell. The membrane is a barrier to the free passage of inorganic ions and most other charged or polar compounds. Transport proteins in the plasma membrane allow the passage of certain ions and molecules. Receptor proteins transmit signals into the cell, and membrane enzymes participate in some reaction pathways. Because the individual lipids and proteins of the plasma membrane are not covalently linked, the entire structure is remarkably flexible, allowing changes in the shape and size of the cell. As a cell grows, newly made lipid and protein molecules are inserted into its plasma membrane. Cell division produces two cells, each with its own membrane. This growth and cell division, fission, occurs without loss of membrane integrity. The internal volume enclosed by the plasma membrane, the cytoplasm, Fig. 1-3, is composed of an aqueous solution, the cytosol, and a variety of suspended particles with specific functions. These particulate components, membranous organelles such as mitochondria and chloroplasts, supramolecular structures such as ribosomes and proteasomes, the sites of protein synthesis and degradation, sediment when cytoplasm is centrifuged at 150,000 g. G is the gravitational force of Earth. What remains as the supernatant fluid is the cytosol, a highly concentrated solution containing enzymes and the RNA molecules that encode them, the components, amino acids, and nucleotides, from which these macromolecules are assembled. Hundreds of small organic molecules called metabolites, intermediates in biosynthetic and degradative pathways, coenzymes, compounds essential to many enzyme-catalyzed reactions, and inorganic ions, K+, Na+, Mg2+, and Ca2+, for example. Of membrane-bounded organelles, including mitochondria, all cells have, for at least some part of their life, either a nucleoid or a nucleus, in which the genome, the complete set of genes, composed of DNA, is replicated and stored, with its associated proteins. The nucleoid in bacteria and archaea is not separated from the cytoplasm by a membrane. The nucleus, in eukaryotes, is enclosed within a double membrane, the nuclear envelope. Cells with nuclear envelopes make up the large domain eukarya, Greek eu, true, and karyon, nucleus. Microorganisms without nuclear membranes, formerly grouped together as prokaryotes, Greek pro, before, are now recognized as comprising two very distinct groups, the domains bacteria and archaea, described below. Cellular dimensions are limited by diffusion. Most cells are microscopic, invisible to the unaided eye. Animal and plant cells are typically 5 to 100 μm in diameter, and many unicellular microorganisms are only 1 to 2 μm long. See the inside of the back cover for information on units and their abbreviations. What limits the dimensions of a cell? The lower limit is probably set by the minimum number of each type of biomolecule required by the cell. The smallest cells, certain bacteria known as mycoplasmas, are 300 nanometers in diameter and have a volume of about 10-14 ml. A single bacterial ribosome is about 20 nanometers in its longest dimension, so a few ribosomes take up a substantial fraction of the volume in a mycoplasmal cell. ml. A single bacterial ribosome is about 20 nanometers in its longest dimension, so a few ribosomes take up a substantial fraction of the volume in a mycoplasmal cell. The upper limit of cell size is probably set by the rate of diffusion of solute molecules in aqueous systems. For example, a bacterial cell that depends on oxygen-consuming reactions for energy extraction must obtain molecular oxygen by diffusion from the surrounding medium through its plasma membrane. The cell is so small, and the ratio of its surface area to its volume is so large, that every part of its cytoplasm is easily reached by O2 diffusing into the cell. With increasing cell size, however, surface-to-volume ratio decreases, until metabolism consumes O2 faster than diffusion can supply it. Metabolism that requires O2 thus becomes impossible as cell size increases beyond a certain point, placing a theoretical upper limit on the size of cells. Oxygen is only one of many low molecular weight species that must diffuse from outside the cell to various regions of its interior, and the same surface-to-volume argument applies to each of them as well. Many types of animal cells have a highly folded or convoluted surface that increases their surface-to-volume ratio and allows higher rates of uptake of materials from their surroundings. Fig. 1-4. Organisms belong to three distinct domains of life. The development of techniques for determining DNA sequences quickly and inexpensively has greatly improved our ability to deduce evolutionary relationships among organisms. Similarities between gene sequences in various organisms provide deep insight into the course of evolution. In one interpretation of sequence similarities, all living organisms fall into one of three large groups, domains, that define three branches of the evolutionary tree of life originating from a common progenitor. Fig. 1-5. Two large groups of single-celled microorganisms can be distinguished on genetic and biochemical grounds: bacteria and archaea. Bacteria inhabit soils, surface waters, and the tissues of other living or decaying organisms. Many of the archaea, recognized as a distinct domain by Carl Woese in the 1980s, inhabit extreme environments: salt lakes, hot springs, highly acidic bogs, and the ocean depths. The available evidence suggests that the archaea and bacteria diverged early in evolution. All eukaryotic organisms, which make up the third domain, eukarya, evolved from the same branch that gave rise to the archaea. Eukaryotes are therefore more closely related to archaea than to bacteria. Model may organisms. Genomic sequences from a wide range of bacteria within the domains of archaea and bacteria are subgroups distinguished by their habitats. In aerobic habitats with a plentiful supply of oxygen, some resident organisms derive energy from the transfer of electrons from fuel molecules to oxygen within the cell. Other environments are anaerobic, devoid of oxygen, and microorganisms adapted to these environments obtain energy by transferring electrons to nitrate, forming into sulfate, forming H2S, or CO2, forming CH4. Many organisms that have evolved in anaerobic environments are obligate anaerobes. They die when exposed to oxygen. Others are facultative anaerobes, able to live with or without oxygen. Organisms differ widely in their sources of energy. And biosynthetic precursors. We can classify organisms according to how they obtain the energy and carbon they need for synthesizing cellular material, as summarized in Fig. 1.6. There are two broad categories based on energy sources: phototrophs, Greek trophy, nourishment, trap and use sunlight, and chemotrophs derive their energy from oxidation of a chemical fuel. Some chemotrophs oxidize inorganic fuels HS to S0, elemental sulfur, S02, or Fe2+ to Fe3+, for example. Phototrophs and chemotrophs may be further divided into those that can synthesize all of their biomolecules directly from CO2, autotrophs, and those that require some preformed organic nutrients made by other organisms, heterotrophs. We can describe an organism's mode of nutrition by combining these terms. For example, cyanobacteria are photoautotrophs, humans are chemoheterotrophs. Even finer distinctions can be made, and many organisms can obtain energy and cyanobacteria are photoautotrophs, carbon from more than one source under different environmental or developmental conditions. Figure 1. Bacterial and archaeal cells share common features, but differ in important ways. The best-studied bacterium, Escherichia coli, is a usually harmless inhabitant of the human intestinal tract. The E. coli cell, Fig. 1-7a, is an ovoid about 2 μm long and a little less than 1 μm in diameter, but other bacteria may be spherical or rod-shaped, and some are substantially larger. E. coli has a protective outer membrane and an inner plasma membrane that encloses the cytoplasm and the nucleoid. Between the inner and outer membranes is a thin but strong layer of a high molecular weight polymer, peptidoglycan, that gives the cell its shape and rigidity. The plasma membrane and the layers outside it constitute the cell envelope. The plasma membranes of bacteria consist of a thin bilayer of lipid molecules penetrated by proteins. Archaeal plasma membranes have a similar architecture, but the lipids can be strikingly different from those of bacteria, see Fig. 10-11. Bacteria and Archaea have group-specific specializations of their cell envelopes, Fig. 1-7bd. Some bacteria, called gram-positive because they are colored by Gram stain, introduced by Hans Peter Gram in 1882, have a thick layer of peptidoglycan outside their plasma membrane, but lack an outer membrane. Gram-negative bacteria have an outer membrane composed of a lipid bilayer into which are inserted complex lipopolysaccharides and proteins called porins that provide transmembrane channels for the diffusion of low molecular weight compounds and ions across this outer membrane. The structures outside the plasma membrane of Archaea differ from organism to organism, but they, too, have a layer of peptidoglycan or protein that confers rigidity on their cell envelopes. Figure 1-7. Diffuse through. The cytoplasm of E. coli contains about 15,000 ribosomes, various numbers, 10 to thousands, of copies of each of 1,000 or so different enzymes, perhaps 1,000 organic compounds of molecular weight less than 1,000, metabolites and cofactors, and a variety of inorganic ions. The nucleoid contains a single, circular molecule of DNA, and the cytoplasm, like that of most bacteria, contains one or more smaller, circular segments of DNA called plasmids. In nature, some plasmids confer resistance to toxins and antibiotics in the environment. In the laboratory, these DNA segments are especially amenable to experimental manipulation and are powerful tools for genetic engineering. See Chapter 9. Other species of bacteria, as well as Archaea, contain a similar collection of biomolecules, but each species has physical and metabolic specializations related to its environmental niche and nutritional sources. Cyanobacteria, for example, have internal membranes specialized to trap energy from light. See Fig. 20-27. Many Archaea live in extreme environments and have biochemical adaptations to survive in extremes of temperature, pressure, or salt concentration. Differences in ribosomal structure gave the first hints that bacteria and Archaea constituted separate domains. Most bacteria, including E. coli, exist as individual cells, but often associate in biofilms or mats, in which large numbers of cells adhere to each other and to some solid substrate beneath or at an aqueous surface. Cells of some bacterial species, the Myxobacteria, for example, show simple social behavior, forming many celled aggregates in response to signals between neighboring cells. Eukaryotic cells have a variety of membranous organelles, which can be isolated for study. Typical eukaryotic cells, Fig. 1-8, are much larger than bacteria, commonly 5 to 100 μm in diameter, with cell volumes 1,000 to 1,000,000 times larger than those of bacteria. The distinguishing characteristics of eukaryotes are the nucleus and a variety of membrane-enclosed organelles with specific functions. These organelles include mitochondria, the site of most of the energy-extracting reactions of the cell, the endoplasmic reticulum and Golgi complexes, which play central roles in the synthesis and processing of lipids and membrane proteins, peroxisomes, in which very long-chain fatty acids are oxidized, and lysosomes, filled with digestive enzymes to degrade unneeded cellular debris. In addition to these, plant cells also contain vacuoles, which store large quantities of organic acids, and chloroplasts, in which sunlight drives the synthesis of ATP in the process of photosynthesis. Fig. 1-8. Also present in the cytoplasm of many cells are granules or droplets containing stored nutrients such as starch and fat. In a major advance in biochemistry, Albert Claude, Christian de Duva, and George Palade developed methods for separating organelles from the cytosol and from each other, an essential step in investigating their structures and functions. In a typical cell fractionation, Fig. 1-9, cells or tissues in solution are gently disrupted by physical shear. This treatment ruptures the plasma membrane, but leaves most of the organelles intact. The homogenate is then centrifuged. Organelles such as nuclei, mitochondria, and lysosomes differ in size and therefore sediment at different rates. These methods were used to establish, for example, that lysosomes contain degradative enzymes, mitochondria contain oxidative enzymes, and chloroplasts contain photosynthetic pigments. The isolation of an organelle enriched in a certain enzyme is often the first step in the purification of that enzyme. The cytoplasm is organized by the cytoskeleton and is highly dynamic. Fluorescence microscopy reveals several types of protein filaments crisscrossing the eukaryotic cell, forming an interlocking three-dimensional meshwork, the cytoskeleton. Eukaryotes have three general types of cytoplasmic filaments: actin filaments, microtubules, and intermediate filaments, Fig. 1 to 10, differing in width from about 6 to 22 nanometers, composition, and specific function. All types provide structure and organization to the cytoplasm and shape to the cell. Actin filaments and microtubules also help to produce the motion of organelles or of the whole cell. Each type of cytoskeletal component consists of simple protein subunits that associate non-covalently to form filaments of uniform thickness. These filaments are not permanent structures. They undergo constant disassembly into their protein subunits and reassembly into filaments. Their locations in cells are not rigidly fixed, but may change dramatically with mitosis, cytokinesis, amoeboid motion, or changes in cell shape. The assembly, disassembly, and location of all types of filaments are regulated by other proteins, which serve to link or bundle the filaments or to move cytoplasmic organelles along the filaments. Bacteria contain actin-like proteins that serve similar roles in those cells. The picture that emerges from this brief survey of eukaryotic cell structure is of a cell with a meshwork of structural fibers and a complex system of membrane-enclosed compartments, Fig. 1 to 8. The filaments disassemble and then reassemble elsewhere. Membranous vesicles bud from one organelle and fuse with another. Organelles move through the cytoplasm. Along protein filaments, their motion powered by energy-dependent motor. Proteins. The endomembrane system segregates specific metabolic processes and provides surfaces on which certain enzyme-catalyzed reactions occur. Exocytosis and endocytosis, mechanisms of transport out of and into cells, respectively, that involve membrane fusion and fission, provide paths between the cytoplasm and surrounding medium, allowing the secretion of substances produced in the cell and uptake of extracellular materials. Figure 1 to 8 Eukaryotic cell structure. This structural organization of the cytoplasm is far from random. The motion and positioning of organelles and cytoskeletal elements are under tight regulation, and at certain stages in its life, a eukaryotic cell undergoes dramatic, finely orchestrated reorganizations, such as the events of mitosis. The interactions between the cytoskeleton and organelles are non-covalent, reversible, and subject to regulation in response to various intracellular and extracellular signals. Figure 1 to 9 Subcellular mechanically aqueous organelles, thus balanced. Figure 1 to 10 The cells build supramolecular structures. Macromolecules and their monomeric subunits differ greatly in size, Fig. 1:11. An alanine molecule is less than 0.5 nanometers long. A molecule of hemoglobin, the oxygen-carrying protein of erythrocytes, red blood cells, consists of nearly 600 amino acid subunits in 4 long chains, folded into globular shapes and associated in a structure 5.5 nanometers in diameter. In turn, proteins are much smaller than ribosomes, about 20 nanometers in diameter, which are much smaller than organelles such as mitochondria, typically 1000 nanometers in diameter. It is a long jump from simple biomolecules to cellular structures that can be seen with the light microscope. Figure 1-12 illustrates the structural hierarchy in cellular organization. Figure 1-11. The monomeric subunits of proteins, nucleic acids, and polysaccharides are joined by covalent bonds. In supramolecular complexes, however, macromolecules are held together by non-covalent interactions much weaker, individually, than covalent bonds. Among these non-covalent interactions are hydrogen bonds, between polar groups, ionic interactions, between charged groups, aggregations of nonpolar groups in aqueous solution brought about by the hydrophobic effect, sometimes called hydrophobic interactions, and van der Waals interactions, also called London forces, all of which have energies much smaller than those of covalent bonds. These non-covalent interactions are described in Chapter 2. The large numbers of weak interactions between macromolecules and supramolecular complexes stabilize these assemblies, producing their unique structures. Figure 1-12. In vitro studies may overlook important interactions among molecules. One approach to understanding a biological process is to study purified molecules in vitro, in glass in the test tube, without interference from other molecules present in the intact cell, that is, in vivo, in the living. Although this approach has been remarkably revealing, we must keep in mind that the inside of a cell is quite different from the inside of a test tube. The interfering components eliminated by purification may be critical to the biological function or regulation of the molecule purified. For example, in vitro studies of pure enzymes are commonly done at very low enzyme concentrations in thoroughly stirred aqueous solutions. In the cell, an enzyme is dissolved or suspended in the gel-like cytosol with thousands of other proteins, some of which bind to that enzyme and influence its activity. Some enzymes are components of multi-enzyme complexes in which reactants are channeled from one enzyme to another, never entering the bulk solvent. When all of the known macromolecules in a cell are represented in their known dimensions and concentrations, Fig. 1-13, it is clear that the cytosol is very crowded, and that diffusion of macromolecules within the cytosol must be slowed by collisions with other large structures. In short, a given molecule may behave quite differently in the cell and in vitro. A central challenge of biochemistry is to understand the influences of cellular organization and macromolecular associations on the function of individual enzymes and other biomolecules to understand function in vivo, as well as in vitro. Figure 1-13. The crowded cell. This drawing by David Goodsell is an accurate representation of the relative sizes. One small region of an E. coli. Summary 1.1 Cellular foundations. All cells are bounded by a plasma membrane, have a cytosol containing metabolites, coenzymes, inorganic ions, and enzymes, and have a set of genes contained within a nucleoid, bacteria and archaea, or nucleus, eukaryotes. All organisms require a source of energy to perform cellular work. Phototrophs obtain energy from sunlight, chemotrophs obtain energy from chemical fuels, oxidizing the fuel and passing electrons to good electron acceptors, inorganic compounds, organic compounds, or molecular oxygen. Bacterial and archaeal cells contain cytosol, a nucleoid, and plasmids, all within a cell envelope. Eukaryotic cells have a nucleus and are multi-compartmented, with certain processes segregated in specific organelles. Organelles can be separated and studied in isolation. Cytoskeletal proteins assemble into long filaments that give cells shape and rigidity and serve as rails along which cellular organelles move throughout the cell. Supramolecular complexes held together by non-covalent interactions are part of a hierarchy of structures, some visible with the light microscope. When individual molecules are removed from these complexes to be studied in vitro, interactions important in the living cell may be lost. 1.2 Chemical foundations. Biochemistry aims to explain biological form and function in chemical terms. By the late 18th century, chemists had concluded that the composition of living matter is strikingly different from that of the inanimate world. Antoine Laurent Lavoisier, 1743 to 1794, noted the relative chemical simplicity of the mineral world and contrasted it with the complexity of the plant and animal worlds. The latter, he knew, were composed of compounds rich in the elements carbon, oxygen, nitrogen, and phosphorus. During the first half of the 20th century, parallel biochemical investigations of glucose breakdown in yeast and in animal muscle cells revealed remarkable chemical similarities between these two apparently very different cell types. The breakdown of glucose in yeast and in muscle cells involved the same 10 chemical intermediates and the same 10 enzymes. Subsequent studies of many other biochemical processes in many different organisms have confirmed the generality of this observation neatly. Summarized in 1954 by Jacques Monod, what is true of E. coli is true of the elephant. The current understanding that all organisms share a common evolutionary origin is based in part on this observed universality of chemical intermediates and transformations, often termed biochemical unity. Fewer than 30 of the more than 90 naturally occurring chemical elements are essential to organisms. Most of the elements in living matter have a relatively low atomic number. Only 3 have an atomic number above that of selenium, 34, Fig. 1 to 14. The 4 most abundant elements in living organisms, in terms of percentage of total number of atoms, are hydrogen, oxygen, nitrogen, and carbon, which together make up more than 99% of the mass of most cells. They are the lightest elements capable of efficiently forming 1, 2, 3, and 4 bonds, respectively. In general, the lightest elements form the strongest bonds. The trace elements represent a minuscule fraction of the weight of the human body, but all are essential to life, usually because they are essential to the function of specific proteins, including many enzymes. The oxygen transporting capacity of the hemoglobin molecule, for example, is absolutely dependent on 4 iron ions that make up only 0.3% of its mass. Biomolecules are compounds of carbon with a variety of functional groups. The chemistry of living organisms is organized around carbon, which accounts for more than half of the dry weight of cells. Carbon can form single bonds with hydrogen atoms, and both single and double bonds with oxygen and nitrogen atoms, Fig. 1-15. Of greatest significance in biology is the ability of carbon atoms to form very stable single bonds with up to 4 other carbon atoms. 2 carbon atoms also can share 2 or 3 electron pairs, thus forming double or triple bonds. The 4 single bonds that can be formed by a carbon atom project from the nucleus to the 4 apices of a tetrahedron, Fig. 1-16, with an angle of about 109.5 degrees between any 2 bonds and an average bond length of 0.154 nm. There is free rotation around each single bond, unless very large or highly charged groups are attached to both carbon atoms, in which case rotation may be restricted. A double bond is shorter, about 0.134 nanometers, and rigid, and allows only limited rotation about its axis. Figure 1-14. Figure 1-15. Figure. Covalently linked carbon atoms in biomolecules can form linear chains, branched chains, and cyclic structures. It seems likely that the bonding versatility of carbon, with itself and with other elements, was a major factor in the selection of carbon compounds for the molecular machinery of cells during the origin and evolution of living organisms. No other chemical element can form molecules of such widely different sizes, shapes, and composition. Most biomolecules can be regarded as derivatives of hydrocarbons, with hydrogen atoms replaced by a variety of functional groups that confer specific chemical properties on the molecule, forming various families of organic compounds. Typical of these are alcohols, which have 1 or more hydroxyl groups, amines, with amino groups, aldehydes and ketones, with carbonyl groups, and carboxylic acids, with carboxyl groups, Fig. 1-17. Many biomolecules are polyfunctional, containing 2 or more types of functional groups, Fig. 1-18, each with its own chemical characteristics and reactions. The chemical personality of a compound is determined by the chemistry of its functional groups and their disposition in 3-dimensional space. Cells contain a universal set of small molecules. Dissolved in the aqueous phase, cytosol, of all cells, is a collection of perhaps 1,000 different small organic molecules, Mr. 100 to 500, with intracellular concentrations ranging from nanomolar to millimolar, see Fig. 15-4, see box 1-1 for an explanation of the various ways of referring to molecular weight. These are the central metabolites and the major pathways occurring in nearly every cell, the metabolites and pathways that have been conserved throughout the course of evolution. This collection of molecules includes the common amino acids, nucleotides, sugars and their phosphorylated derivatives, and mono-, D-, and tricarboxylic acids. The molecules may be polar or charged and are water-soluble. They are trapped in the cell because the plasma membrane is impermeable to them, although specific membrane transporters can catalyze the movement of some molecules into and out of the cell or between compartments in eukaryotic cells. The universal occurrence of the same set of compounds in living cells reflects the evolutionary conservation of metabolic pathways that developed in the earliest cells. Box 1-to-1 molecular. There are two of an atom of carbon. Figure 1-to-17. There are other small biomolecules specific to certain types of cells or organisms. For example, vascular plants contain, in addition to the universal set, small molecules called secondary metabolites, which play roles specific to plant life. These metabolites include compounds that give plants their characteristic scents and colors, and compounds such as morphine, quinine, nicotine, and caffeine that are valued for their physiological effects on humans, but have other purposes in plants. Figure 1. The entire collection of small molecules in a given cell under a specific set of conditions has been called the metabolome, in parallel with the term genome. Metabolomics is the systematic characterization of the metabolome under very specific conditions, such as following administration of a drug or a biological signal such as insulin. Macromolecules are the major constituents of cells. Many biological molecules are macromolecules, polymers with molecular weights above 5,000, that are assembled from relatively simple precursors. Shorter polymers are called oligomers, Greek oligos, few. Proteins, nucleic acids, and polysaccharides are macromolecules composed of monomers with molecular weights of 500 or less. Synthesis of macromolecules is a major energy-consuming activity of cells. Macromolecules themselves may be further assembled into supramolecular complexes, forming functional units such as ribosomes. Table 1-to-1 shows the major classes of biomolecules in an E. coli cell. Proteins, long polymers of amino acids, constitute the largest fraction, besides water, of a cell. Some proteins have catalytic activity and function as enzymes. Others serve as structural elements, signal receptors, or transporters that carry specific substances into or out of cells. Proteins are perhaps the most versatile of all biomolecules. A catalog of their many functions would be very long. The sum of all the proteins functioning in a given cell is the cell's proteome, and proteomics is the systematic characterization of this protein complement under a specific set of conditions. The nucleic acids, DNA and RNA, are polymers of nucleotides. They store and transmit genetic information, and some RNA molecules have structural and catalytic roles in supramolecular complexes. The genome is the entire sequence of a cell's DNA, or in the case of RNA viruses, its RNA, and genomics is the characterization of the structure, function, evolution, and mapping of genomes. The polysaccharides, polymers of simple sugars such as glucose, have three major functions: as energy-rich fuel stores, as rigid structural components of cell walls, in plants and bacteria, and as extracellular recognition elements that bind to proteins on other cells. Shorter polymers of sugars, oligosaccharides, attached to proteins or lipids at the cell surface serve as specific cellular signals. A cell's glycome is its entire complement of carbohydrate-containing molecules. The lipids, water-insoluble hydrocarbon derivatives, serve as structural components of membranes, energy-rich fuel stores, pigments, and intracellular signals. The lipid-containing molecules in a cell constitute its lipidome. With the application of sensitive methods with great resolving power, mass spectrometry, for example, it is possible to distinguish and quantify hundreds or thousands of these components, and thus to quantify their variations in response to changing conditions, signals, or drugs. Systems biology is an approach that tries to integrate the information from genomics, proteomics, and metabolomics to give a molecular picture of all the activities of a cell under a given set of conditions and the changes that occur when the system is perturbed by external signals or circumstances, or by mutations. Table 1 to Table 1 Cell Percent Approx Water Protein D RNA Poly Lipid Mon 22 Inorganic Source Proteins, polynucleotides, and polysaccharides have large numbers of monomeric subunits, and thus high molecular weights in the range of 5,000 to more than 1 million for proteins, up to several billion for nucleic acids, and in the millions for polysaccharides such as starch. Individual lipid molecules are much smaller, Mr. 750 to 1,500, and are not classified as macromolecules, but they can associate non-covalently into very large structures. Cellular membranes are built of enormous non-covalent aggregates of lipid and protein molecules. Given their characteristic information-rich subunit sequences, proteins and nucleic acids are often referred to as informational macromolecules. Some oligosaccharides, as noted above, also serve as informational molecules. Three-dimensional structure is described by configuration and conformation. The covalent bonds and functional groups of a biomolecule are, of course, central to its function, but so also is the arrangement of the molecule's constituent atoms in three-dimensional space, its stereochemistry. Carbon-containing compounds commonly exist as stereoisomers, molecules with the same chemical bonds and same chemical formula, but different configuration, the fixed spatial arrangement of atoms. Interactions between biomolecules are invariably stereospecific, requiring specific configurations in the interacting molecules. Figure 1 to 19 representations of molecules. 3 ways to represent the structure of the amino acid alanine, shown here in the ionic form found at neutral. Figure 1 to 19 shows 3 ways to illustrate the stereochemistry, or configuration, of simple molecules. The perspective diagram specifies stereochemistry unambiguously, but bond angles and center-to-center bond lengths are better represented with ball and stick models. In space-filling models, the radius of each atom is proportional to its van der Waals radius, and the contours of the model define the space occupied by the molecule, the volume of space from which atoms of other molecules are excluded. Configuration is conferred by the presence of either 1. Double bonds, around which there is little or no freedom of rotation, or 2. Chiral centers. around which substituent groups are arranged in a specific orientation. The identifying characteristic of stereoisomers is that they cannot be interconverted without temporarily breaking one or more covalent bonds. Figure 1-20 shows the configurations of maleic acid and its isomer, fumaric acid. These compounds are geometric isomers or cis-trans isomers. They differ in the arrangement of their substituent groups with respect to the non-rotating double bond, Latin cis, on this side groups on the same side of the double bond, trans, across groups on opposite sides. Maleic acid, maleate at the neutral pH of cytoplasm, is the cis isomer, and fumaric acid, fumarate, the trans isomer. Each is a well-defined compound that can be separated from the other, and each has its own unique chemical properties. A binding site on an enzyme, for example, that is complementary to one of these molecules would not be complementary to the other, which explains why the two compounds have distinct biological roles despite their similar chemical makeup. In the second type of stereoisomer, for different substituents bonded to a tetrahedral carbon atom may be arranged in two different ways in space, that is, have two configurations yielding two stereoisomers that have similar or identical chemical properties but differ in certain physical and biological properties. A carbon atom with four different substituents is said to be asymmetric, and asymmetric carbons are called chiral centers, Greek kairos, hand. Some stereoisomers are related structurally as the right hand is to the left. A molecule with only one chiral carbon can have two stereoisomers. When two or more N chiral carbons are present, there can be two N stereoisomers. Stereoisomers that are mirror images of each other are called enantiomers, Fig. 1-21. Pairs of stereoisomers that are not mirror images of each other are called diastereomers, Fig. 1-22. Figure 1-20. When looking down the vertical, Figure 1-20. As Louis Pasteur first observed in 1843, box 1-2, enantiomers have nearly identical chemical reactivities, but differ in a characteristic physical property, their interaction with plane polarized light. In separate solutions, two enantiomers rotate the plane of plane polarized light in opposite directions, but an equimolar solution of the two enantiomers, a racemic mixture, shows no optical rotation. Compounds without chiral centers do not rotate the plane of plane polarized light. Box 1. Veritas. Louis dextrorotator. In isomer, we know Louis. Now we. Figure 1. Pasteur separate. Asterisk. Key convention. For naming in the RS system, the chiral atom is viewed with the group of lowest priority, for in the following diagram, pointing away from the viewer. If the priority of the other three groups, 1-3, decreases in clockwise order, the configuration is R, Latin rectus, right. If counterclockwise, the configuration is S, Latin sinister, left. In this way, each chiral carbon is designated either R or S, and the inclusion of these designations in the name of the compound provides an unambiguous description of the stereochemistry at each chiral center. Another naming system for stereoisomers, the D and L system, is described in Chapter 3. A molecule with a single chiral center, the two isomers of glyceraldehyde, for example, can be named unambiguously by either system, as shown here. Distinct from configuration is molecular conformation, the spatial arrangement of substituent groups that, without breaking any bonds, are free to assume different positions in space because of the freedom of rotation about single bonds. In the simple hydrocarbon ethane, for example, there is nearly complete freedom of rotation around the C-C bond. Many different, interconvertible conformations of ethane are possible, depending on the degree of rotation Fig. 1 to 23. Two conformations are of special interest, the staggered, which is more stable than all others and thus predominates, and the eclipsed, which is the least stable. We cannot isolate either of these conformational forms, because they are freely interconvertible. However, when one or more of the hydrogen atoms on each carbon is replaced by a functional group that is either very large or electrically charged, freedom of rotation around the C-C bond is hindered. This limits the number of stable conformations of the ethane derivative. Figure 1 to 23. Interactions between biomolecules are stereospecific. When biomolecules interact, the fit between them must be stereochemically correct. The three-dimensional structure of biomolecules large and small, the combination of configuration and conformation is of the utmost importance in their biological interactions. Reactant with its enzyme, hormone with its receptor on a cell surface, antigen with its specific antibody, for example, Fig. 1 to 24. The study of biomolecular stereochemistry, with precise physical methods, is an important part of modern research on cell structure and biochemical function. Figure 1 to 2. In living organisms, chiral molecules are usually present in only one of their chiral forms. For example, the amino acids in proteins occur only as their L isomers, glucose occurs only as its D isomer. The conventions for naming stereoisomers of the amino acids are described in Chapter 3, those for sugars in Chapter 7. The RS system, described above, is the most useful for some biomolecules. In contrast, when a compound with an asymmetric carbon atom is chemically synthesized in the laboratory, the reaction usually produces all possible chiral forms, a mixture of the D and L forms, for example. Living cells produce only one chiral form of a biomolecule, because the enzymes that synthesize that molecule are also chiral. Stereospecificity, the ability to distinguish between stereoisomers, is a property of enzymes and other proteins and a characteristic feature of biochemical interactions. If the binding site on a protein is complementary to one isomer of a chiral compound, it will not be complementary to the other isomer, for the same reason that a left glove does not fit a right hand. Some striking examples of the ability of biological systems to distinguish stereoisomers are shown in Figure 1 to 25. The common classes of chemical reactions encountered in biochemistry are described in Chapter 13, as an introduction to the reactions of metabolism. Summary 1.2 Chemical Foundations. Because of its bonding versatility, carbon can produce a broad array of carbon skeletons with a variety of functional groups. These groups give biomolecules their biological and chemical personalities. A nearly universal set of about 1,000 small molecules is found in living cells. The interconversions of these molecules in the central metabolic pathways have been conserved in evolution. Proteins and nucleic acids are linear polymers of simple monomeric subunits. Their sequences contain the information that gives each molecule its three-dimensional structure and its biological functions. Molecular configuration can be changed only by breaking covalent bonds. For a carbon atom with 4 different substituents, a chiral carbon, the substituent groups can be arranged in 2 different ways, generating stereoisomers with distinct properties. Only one stereoisomer is biologically active. Molecular conformation is the position of atoms in space that can be changed by rotation about single bonds, without breaking covalent bonds. Interactions between biological molecules are almost invariably stereospecific. They require a close fit between complementary structures in the interacting molecules. 1.